Kingdom Protista. Protists are unicellular eukaryotes. The term Protista was first used by Ernst Haeckel in 1866. Protists were traditionally subdivided into several groups based on similarities as chrysophytes, dinoflagellates, euglenoids and slime molds. Protists are either unicellular, or they are multicellular without specialized tissues. This simple cellular organization distinguishes the protists from other eukaryotes, such as fungi, animals and plants. Protists live in almost any environment that contains liquid water. Many protists, such as algae, are photosynthetic and are vital primary producers in ecosystems, particularly in the ocean as part of the plankton. Other protists, such as the kinetoplastids and epicompalgs, are responsible for a range of serious human diseases, such as malaria and sleeping sickness. As we recollect, Protista is subdivided into chrysophytes, dinoflagellates, euglenoids and slime molds. Let us first learn about chrysophytes. The chrysophytes, also known as golden algae, are a large group of heterocont algae, found mostly in fresh water. Chrysophytes are mostly unicellular flagellates, with either two visible flagella, example, Ochromonas, or sometimes one flagella, example, Cromulina. Some members are generally amoeboid, with long branching cell extensions, example, Chrysamoeba and Rhizochrysis. Diatoms are a major group of chrysophytes, also known as eukaryotic algae, and are one of the most common types of phytoplankton. A characteristic feature of diatom cells is that they are encased within a unique cell wall, made of silica, which is hydrated silicon dioxide and is called a frustule. These frustules show a wide diversity in form, some quite beautiful and ornate, but usually consist of two asymmetrical sides with a split between them, hence the group name. Fossilized remains of diatoms over 1 billion of years forms diatomaceous earth. The next group of Protista are dinoflagellates. The dinoflagellates are a large group of flagellate protists. Most are marine plankton, but they are common in freshwater habitats as well. Their populations are distributed depending on temperature, salinity, or depth. About half of all dinoflagellates are photosynthetic. Most dinoflagellates are unicellular forms with two flagella. One of these extends towards the posterior, called the longitudinal flagellum, while the other forms a lateral circle, called the transverse flagellum. Red tide is more specifically produced when dinoflagellates are able to reproduce rapidly and copiously on account of the abundant nutrients in the water. Although the resulting red waves are a miraculous sight, they, again, contain toxins that not only affect all marine life in the ocean but the people who consume them as well. A specific carrier is shellfish. This can introduce both non-fatal and fatal illness, due to the toxins they produce. The next group of protists are euglenoids. The euglenoids were first defined by Otto Büchli in 1884 as the flagellate order Euglenida. Botanists subsequently treated the algal division Euglenophyta, thus they were classified as both animals and plants, as they share characteristics with both. The euglenoids are one of the best-known groups of flagellates, commonly found in freshwater, especially when it is rich in organic materials. Example, Euglena. Many euglenoids have chloroplasts and produce energy through photosynthesis, but others feed by phagocytosis or strictly by diffusion. Euglenoids are distinguished mainly by the presence of a pellicle, which is composed of proteinaceous strips underneath the cell membrane, supported by dorsal and ventral microtubules. This varies from rigid to flexible, and gives the cell its shape, often giving it distinctive striations. In many euglenoids the strips can slide past one another, causing an inching motion called metaboly. Otherwise they move using the flagella. The last group of protists are the slime molds. Slime molds feed on microorganisms in decaying vegetable matter. They can be found in the soil, on lawns, and in the forest, commonly on deciduous logs. They are also common on mulch, or even in leaf mold, which inhabits stitches. They begin life as amoeba-like cells. Slime molds are commonly haploid and multiply if they encounter their favorite food, bacteria. These amoebae can mate if they encountered the correct mating type and form zygotes, which then grow into plasmodia, which contain many nuclei without cell membranes between them, which can grow to be meters in size. When the food supply weans, the plasmodium will migrate to the surface of its substrate and transform into rigid fruiting bodies. The fruiting bodies, or sporangia, are what we commonly see, superficially look like fungi or molds, but they are not related to the true fungi. These sporangia will then release spores which hatch into amoebae to begin the life cycle again.